Tony.
Very nice to meet you as well. Thank you very much for having me. Hello. In this talk, we're going to ask two questions, really. What is heritable human genome editing, and when will it happen? And the way we're going to do this is, first of all, in the next 15 minutes or so, I'll try and define some of the terms, give them the meanings. I'll try and explain a little bit about how genome editing works. I'll introduce some characters along the way, some fictional characters to try and illustrate this, and right at the end we'll finish with a summary. So to begin with the first definition, by edit, we simply, because we're editing the genome, by edit we simply mean the standard dictionary definition of edit, which is to make a precise and controlled change. And that change can be a very large-scale change in genome terms or a very small change. But what it is that we're editing really is the genome, and the genome can be thought of as really like an instruction manual. And most of the trillions of cells in the bodies of each of us contain two instruction manuals, one that we've inherited from our mother and one that we've inherited from our father. And each one of those instruction manuals contains 3.2 billion characters or so, making a total of 6.4 billion characters per cell. And to give you an idea of how many that is, if we imagine that each character is a lowly ping pong ball, there'd be enough ping pong balls to fill Wembley Stadium, which is the national soccer stadium of England, not once, but 6 times. So as you can see, I'm talking a lot of balls. So if you want to go back and think about the history of us being able to make a change, perhaps even a change to just one of those 6 billion in the entire instruction manual, before 1989, the history looked like this. There wasn't any history at all, we simply couldn't do it. And then the work of these 3 geniuses was pulled together, and they won the Nobel Prize for this in 2007, which gave us a method of editing, of making edits or prescribed changes, mainly in mice, and this has been extremely powerful for biomedical research, but it's clunky. It's slow, it takes at least a year in some cases, it's limited to one target at a time, it's very expensive, it's pricey, and there are all sorts of other constraints as well. And so the search for alternatives continued, and in 2005 and 2010 it was rewarded with the discovery of zinc finger nucleases, ZFNs, and then talons. You might have heard of these, and these are fantastic as well, but there's still no cigar, and the search continued. And then, in 2012 and 2013, with these 3 papers in Science magazine, the CRISPR-Cas9 revolution began and was announced. This new method of editing genomes. And unlike its predecessors, editing by CRISPR-Cas9 is extremely fast. It takes weeks, not up to a year or more. You can multiplex, so you can target multiple targets in one shot. It's economical. It costs hundreds of dollars, not thousands or tens of thousands of dollars. It's streamlined. It's already widely practiced, and it is highly efficient. You shoot, you score. So although CRISPR-Cas9 editing doesn't do anything that we couldn't do before, it just does it so much better that it's a game changer. So how does it work? Well, it works a little bit like this in 2 steps, and the CRISPR-Cas9 plays the part in the first step, and then the second step is taken over by the cell. And I'll try and explain this one by one, starting with CRISPR-Cas9. And CRISPR-Cas9 is really like a tiny pair of molecular scissors and a molecular sat-nav. So like all sat-navs, you get to program whereabouts you want to go, not on a map this time, but in the genome. And in fact, the molecule that's responsible for this, the nature of the molecule that's the sat-nav is what's called an RNA molecule. Sometimes you see it prefixed by a little g or a little s, single guide RNA or guide RNA. But that's the sat-nav, and it works by carrying the scissors to the precise place that you want it to go to on the genome. This is how it works in a very kind of simplified diagram. The central part of this is a part of the genome, the 6.4 billion characters. And you can see at the bottom, I've got let's see if I can get this to work. Here, this is the sat-nav, the guide RNA, and it looks incredibly prosaic, but in fact it's amazing because it can combine with 2 different entities. One of them is the pink blobby thing here, which is the molecular scissors, the Cas9, and you can see it interacting here, but the other part of the interaction is just 20 characters long, and it causes a specific interaction with the genome, those 6.4 billion characters. And you might think, well, there's only 20 characters here that are matching up. Surely that's going to stick everywhere on the 6.4 billion. It's not going to be nearly specific enough. And you'd be quite wrong. 20 characters is more than enough to endow exquisite specificity to this system. So So now the CRISPR-Cas9 has made a cut in the genome. We're switching similes here. So now, like a piece of string, we've made a targeted cut exactly where we want it to be. But actually, cuts like this, breaks in the genome, can occur from time to time naturally, and they are very bad news, and they have to be repaired by the cell, and very rapidly, so that if we look at how that happens, in our case, we've made the break using CRISPR-Cas9, targeted to a particular place. This is all taking place inside the cell, and then the cell uses its repair machinery like this, to fix that break. And I think of this repair machinery, this is homing in really on what's going on, as being a bit like a bridge, a molecular bridge. Of course, this is all happening in tiny, tiny scale, where 1 foot of the bridge, the left foot of the bridge, perfectly matches the left-hand side of the break, and the right foot of the bridge perfectly matches the right side of the break. But the span of the bridge, you get to put in whatever you like. So this is an extra, just a little piece of the genome, of the instruction manual. You can flood the system with this, and you've figured out exactly what you want to do so that it ends up being integrated into the genome. So you've made this prescribed change. And what this enables us to do is put in either very, very small changes, in fact they can reflect a small deletion, or huge changes of many thousands of characters. And this really makes me think that we've given a carpenter, a kind of molecular carpenter, an amazing new set of tools. And of course, like all carpenters, he or she thinks what they're going to make, so they have a vision that they want to realize, and they bring their experience and expertise and other knowledge to apply the tools to realize the vision. So I've spent a little bit of time talking about the tools, the CRISPR-Cas9, but what about the vision? Well, most people think that one of the major applications for CRISPR-Cas9 editing will be in human clinical medicine. And to illustrate this, I'd like to introduce you to Trubshaw. Trubshaw was born at 7:42 a.m. on June the 14th, 2013. Happy birthday, Trubshaw. By the time he's 17, he'll be a multi-billionaire, and at age 28, he'll go on to found Trubshaw Lunar Mining Inc. But when he's 34, he'll be diagnosed with an inherited terminal neurodegenerative disease. What hope might there be in 2047 for Trubshaw and people like him? Well, I think that we'll be able to combine different technologies, including CRISPR-Cas9 and induced pluripotency, to help solve the problem. So Trubshaw will go to his clinic or hospital, perhaps, and give a cheek cell sample, which will then be cultured in the test tube, and caused to become IPS cells, induced pluripotent cells. These cells are like embryonic cells, but they're not embryos, and so they've kind of like their developmental tabula rasa has been erased, and they can become anything that we want them to within reason, and I'll explain that a little bit in a minute. But also, because they're proliferating, we can add the editing tools so that now we can edit the gene that was predisposing to Trubshaw's neurodegenerative condition. We can, by a process called directed differentiation, then ask these cells to become neurons, and since they kind of, in a way, derived from Trubshaw in the first place, we can then transplant them back to Trubshaw so that he is now fixed and better. Hurrah! So he's well, and he's a multi-billionaire, of course, and the ladies love him. And so out he goes into the outside world, and he meets the love of his life, and they get hooked up and married and everything, and they want to start a family. But the problem is that we've fixed Trubshaw's neurons, but it means that any kids that they have may inherit the same condition that Trubshaw Sr. had. So what hope might there be in the future that we can fix that problem? And that's what brings us to the area of heritable human genome editing. And by that, what we mean is where we're making an edit that is passed to successive, or maybe passed to successive generations. And we can do this via one of two hypothetical strategies. Today, they're hypothetical in humans. We're putting the pieces of this jigsaw together in experimental systems, particularly living mice, which is a very important model. But in humans, it's hypothetical. And the two strategies that we think that we will be able to use are to be able to do this in embryos and to be able to do this in gametes. They're both very important, I think, and I'd like to talk about them one by one, starting with editing in embryos. So this is a micrograph of a one-cell mouse embryo. One-cell human embryos look very similar. And it's being injected with a very gentle needle here with the CRISPR-Cas9 components so that they can do the edit of the affected gene at the one-cell stage. And what that means is that then if we now transplant the embryo, if it's Trubshaw's embryo, perhaps into Mrs. T, and she carries the pregnancy, now all of the cells of Trubshaw Jr. when he's born contain that edit, so he will not be predisposed to the same heritable condition. And this, in fact, probably applies to many, many different so-called single-gene diseases, where probably about 6,000 to 8,000, maybe a few handful more are known, where they severally, so individually, they're quite rare, but together, collectively, these single gene diseases, conditions, really add up to something. And in the UK, it's probably around 1 million people are affected. So an enormously important problem. But in fact, all disease probably involves many different genes. And you can think of this in terms of all of the major killers in Western economies and elsewhere. You probably recognize some of them here. Heart disease, chronic heart disease, obesity, diabetes, certain kinds of cancer, or all kinds of cancer. So an enormously important problem. There's a genetic component, an environmental component, and the interactions are phenomenally complicated. And so for this reason, and with the genetic contribution alone being so complicated, we've overlooked the problem because it's just too difficult. But that's probably about to change, and it's about to change with one technology in particular, and it's this: whole genome sequencing. So here is a chart which shows the progress of whole genome sequencing of the human genome since the first publication in 2001. So all, sort of 6.4 billion characters were sequenced for the first time, and the manuals was published, as it were, in 2001 at a cost of approximately $100 million, US dollars. And if we follow the trajectory, sorry, I've jumped the gun a bit here, but if we follow the trajectory of where the cost, which is shown by this green line here, would have been going, according to Moore's law. So Gordon Moore is one of the co-founders of Intel, and in 1965, Gordon Moore published this paper where he predicted that the number of transistors on an integrated circuit would more or less double every year. And there are many iterations of Moore's law now, and if our situation, the cost of genome sequencing, had followed Moore's law, by the end of 2015, it would cost just under about $1 million US dollars. In fact, in the last 10 years, as you can see, the cost has been dropping much more than that on average, 80% per year. And what that means is if we follow the same trajectory cost-wise, in 5 years' time, it will be possible to sequence your genome for 25 cents. So what you'll be able to do is you'll be able to go to your supermarket that we heard about just now, and Moses talking about this somewhat presciently, and you'll be able to go to the whole genome sequencing counter, give her an epithelial swab from your cheek cell, okay? Off it goes in the drone, so they take it away to the sequencing center where your genome will be sequenced, and then it'll be sent up to the cloud for processing, and the processing will be sent to your genetic counselor or your insurer or your healthcare provider to decide what to do with this analysis. And what will this mean? Well, nobody really knows, but I think there are several ways that this might play out. One is that we'll identify new disease genes, because these data will come with patient data as well. And not just new genes, but also combinations of genes, not just in disease, but in health. Great-great-grandmother, smoked sort of 80 Capstan full strengths before breakfast, but lived to be 115. Well, we'll find out why. She survived, and she was well. So it's for disease and health. But another corollary of this, I think, is that it's going to show that every embryo that's produced will have multiple so-called problem genes. So might genome editing in the future be able to fix these multiple problems? And to answer this, I'd like to return to our two editing methods. And before, remember, we were talking about editing the embryo genome, the single-cell embryo genome. And the problem with making multiple edits in a single-cell embryo genome is that the single-cell embryo quickly becomes a two-cell embryo. You want to get at the one-cell embryo, so you've got quite a finite window of opportunity. And if you're doing multiple cuts, there is a risk that you might end up with scrambled eggs. So we don't want that, and I think it's much more likely that this other method, gamete genome editing, will be the one that we choose. And what we're doing here, so the gametes, the sperm or the eggs, and what we're going to be talking about here is, for example, for Mr. T, he'll go along, we'll generate iPS cells from him, and we'll be able to make an edit. and confirm that the edit is exactly what we want, and then make another edit, because we have a much broader window, much wider window of opportunity, and then make multiple sequential rounds of editing in series, confirm each time that we've got what we want and nothing else, and then cause these iPS cells to differentiate not into neurons like before, but into sperm cells that we can then, by using assisted reproductive technologies that are commonplace around the world, inject these sperm into Mrs. T's oocytes, and then she carries the pregnancy, so that now Trubshaw Jr. has multiple edits on his father's side that might fix some of the diseases to which he would otherwise, or she would otherwise have been predisposed. And the same is true for Mrs. T, because probably Mrs. T carries some of these so-called problem genes as well, and we can do exactly the same type of approach, except this time turn them into oocytes or eggs, and then combine them together so that Trubshaw Jr. has edits on both sides. So now this really has given the carpenter a fantastically powerful toolkit. Fantastic tools, and he or she has this vision. But the problem is that the vision of the carpenter and you or I might not be quite the same as Trubshaw's vision. Trubshaw might not be satisfied with sorting out the predisposing genes that cause disease, might predispose to disease. He might have a vision where he wants to create a family of Lara Crofts and Jason Bournes. Right? And so this leads us now into the area of enhancement, a kind of transhumanism light, where we're altering traits that are not necessarily directly related to disease. This leads to the idea of designer babies, and it can be extended, of course, even further, because we could use genome editing to introduce more fantastic, or realize more fantastic visions, where we're introducing entirely new traits that aren't present in the human race at the moment, as far as we know. Want your kids to be able to fly to school in the morning, madam? Not a problem. We'll give them wings. Okay? So there is a problem with this. Okay, there are several problems with it, really. But the problem that I have in mind is that we're forgetting the due process. So the carpenter in his or her workshop must know which genes to edit in order to realize the vision. So this is exactly the same problem, I think, in a different, not exactly the same, it's the kind of problem that also beset Charles Babbage. Charles Babbage was born in 1791, and he's credited with being one of the pioneers of the modern mechanical calculator, the computer. He called it a difference engine. But more than once in his lifetime, he was asked the question, Mr. Babbage, if I put the wrong value into your calculating machine, will it still give me the right answer? Now, we can laugh at this question in a way, but I think it's exactly the same mistake that we're making when we consider genome editing, because we must know exactly which pieces, which genes, which regions of the manual to change. We probably do, in some cases, some single gene conditions that I've mentioned, but for complex traits, we almost certainly don't. And unless we do, we cannot reliably get the right answer. So if we return now, at the end of this talk, to the questions that we asked right at the beginning, what is heritable human genome editing, and when will it happen? I think we can say, in summary, that it involves prescribed changes in the human genome, the instruction manuals that we can perform in early-stage embryos or gametes, sperm and the egg, such that they can be transmitted, they're heritable, transmitted across generations. And probably initially this will involve CRISPR-Cas9 because it's so fantastic, but who knows what technology is around the corner? There might be something else that can do the job even better. When will this happen? Well, certain technologies like sequencing and induced pluripotency are likely to drive the demand for human heritable genome editing. If you know that you've got certain genes that might predispose to disease, why would you want to give those and pass those on to your children? But on the other hand, and pulling in the opposite direction, there are other technical, legislative, political, and social considerations and concerns that will delay human heritable genome editing, pushing it perhaps into the future by years or even decades. Thank you very much indeed.
It's interesting, Tony, that you mention anything remotely resembling eugenics and people run for the doors. It has a very negative heritage. And yet, as you say, why would you pass on something that you know to be damaging? And yet then again, we have many biographies of people who were theoretically damaged and who've gone on to contribute great things to humanity. This is a long discussion about to continue.
I agree.
Yeah. Thank you so much for your
Thank you,
presentation.
Moses. Thank you very much. Thank you.